A flower, sometimes known as a bloom or blossom, is the reproductive structure found in flowering plant. The biological function of a flower is to facilitate reproduction, usually by providing a mechanism for union of sperm and egg. Something that matured is fully developed and is peak of being alive and matured. The major stage of the flower life cycle are the seed, germination, growth, reproduction, pollination, and seed spreading stage.
They have a several characteristics of flower, like distinctive in color and form, the reproduction of the species by seed production, and the flower is made up of a flowering axis. Plus, they have both fundamental reproduction organs and supplementary reproductive organs. Next, the ovary contains the ovule or potential seeds. It might be radially or bilaterally symmetrical. Lastly, the fertilization occurs when pollen grains are transported from anther to a pistil stigma. In genetics, narrow sense heritability has power of 2 and on phenotypic mean basic average over replications and years was calculated using the formula that published by Nguyen and Slipper in the year of 1983. In genetics, the narrow sense heritability formula is used to determine the heritability of a trait due to the additive effect of alleles. An inheritance of summer dormancy, persistence, and productivity uses half-ship mating in tall fescue in addition to the formula. Third fescue researcher has paid less attention to the genetic basis and variability of persistent and summer dormancy, as well as their association with forest production qualities. As a result, considerable genotypic variance was identified in both parental genotypes and half-seed families for all examined variables. The majority of the genotypes tested exhibited insufficient summer dormancy. To summarize, knowing the inheritance and nature of genetic variability, as well as the relationship between features, is crucial for increasing the success of breeding programs and selecting superior genotypes. To collect quantitative genetic information, most forage grasses use half-seed mating, such as top cross, polycross, and open pollination.
The phenotype can be divided into a limited number of readily distinguishable discrete classes when it comes to any of these traits. Such traits are usually controlled by more than one gene. These are called complex traits, for example, height and metabolism rate. Quantitative genetics is important for a variety of reasons. The most of key feature that plant and animal breeder examine are quantitative traits. Quantitative characteristics are frequently measured and described quantitatively, and the measurement of quantitative traits is essential when comparing individuals or evaluating group of individuals. Several genes impact several human illness. Many of the quantitative traits that allow a species to adapt to its environment are present quantitative inheritance, character inheritance that is based on cumulative activity of several genes, such as of which has a small effect. Example of such floral maturity.
This is the list of referring that we use to refer to preparing this slide and presentation. Thank you for being with us to the end.